All right, hi, I'm Alexa. I'm a variant scientist from the Clinical Genomics Laboratory here at Washington University in St. Louis. Thank you so much to the organizers for having me and letting me talk to you about our work in classifying variants associated with a cancer-adjacent topic, disorders of somatic mosaicism. So let's start off with what is somatic mosaicism. So if in development, a genetic variation is introduced after the zygotic stage, only some tissues will carry the variant, not all. And due to this differential tissue expression, as well as due to the timing of the variant introduction, this can result in a wide array of different clinical presentations. And importantly, some of these disorders are treatable, so getting the variant classification right on these is really important. But the issue is that there really are no classification guidelines for these types of variants. We all know that there are guidelines for classifying variants associated with cancer, but again, this is cancer adjacent. There are guidelines for classifying variants associated with germline disorders, but this is also germline adjacent as well. The Clinical Genome Resource, or ClinGen, has started to form expert panels that have begun to specify the 2015 ACMG guidelines for different genes and different disorders. One of the ClinGen expert panels has decided to focus on a very specific disorder of somatic mosaicism in brain malformations. This is the closest guideline that we have to very general disorders of somatic mosaicism. So this is where we decided to start to build our framework for more generally classifying all disorders of a somatic mosaicism. Just a reminder, this is the 2015 ACMG AMP guidelines, and this is what the brain malformations used as their criteria to start to build their specific criteria. And this is the ways in which they modified these criteria to be more specific to variants in AKT3, mTOR, PIK3CA, or PIK3R2, or more specific to brain malformations. You can see that there were 12 criteria that were excluded, either due to updated recommendations or due to these criteria not being applicable for the genes they focused on or for somatic mosaicism. There were 8 criteria that they more specifically defined to be specific for brain malformations or specific for their genes that they focused on, AKT3, mTOR, PIK3CA, or PIK3R2. So how do these guidelines work in classifying a variant that is not in one of those genes and not associated with brain malformations? So here's an example. This is a nonsense variant in the TEC gene, which encodes a receptor tyrosine kinase that's specifically expressed in endothelial cells. Pathogenic TEC variants are associated with venous malformations, and we detected this variant at a reduced variant allele fraction in an individual with a venous malformation. The TEC gene is associated with a gain of protein functionality, and the brain malformations VSEP has said that PVS1 is not applicable, so we did not apply PVS1. This variant has been described in the literature in an individual with a vascular malformation, but under the brain malformations guidelines, this is not applicable under any of their codes. There is a functional study showing increased protein functionality, which is applicable under the PS3 code, but due to updated recommendations, it's only applicable at the moderate level. The variant is also absent from population databases, applicable under PM2, but again, only under the supporting level. And really, there is no extra information on this variant. So according to the brain malformations VSEP guidelines, this variant would be classified as a variant of uncertain significance. But you can see in the C terminus of this protein, there are several truncating pathogenic or likely pathogenic variants that have been detected in individuals with venous malformations. So this probably is not the right classification for this variant. So we needed updated criteria to not only classify this variant, but to more generally classify any variant in a disorder of somatic mosaicism. And the reason that we at the Clinical Genomics Laboratory at Washington University in St. Louis are so uniquely positioned to create such a framework is because our laboratory has been doing this type of testing for a number of years on a large number of samples. Our assay is designed to detect a large variety of disorders of somatic mosaicism. We sequence 129 genes, And we sequence at a very high coverage to be able to detect these variants that may be expressed at a low variant allele fraction in diseased tissues. So back to the brain malformations modified guidelines, these are the framework that we created as compared to the brain malformations VSEP. You can see there were 5 criteria that were excluded that we decided not to use, again, either due to updated recommendations or due to these criteria not being applicable for disorders of somatic mosaicism. 5 criteria were included in our framework that had been previously excluded by the brain malformations VSEP. There were 4 criteria that we differently defined to be able to make these criteria, again, more generally applicable to all disorders of somatic mosaicism. And there was 1 new criterion that we added, which I'll touch upon later. But first, I wanted to focus on 2 criteria that the brain malformations VSEP had excluded in PP3 and BP4, the use of in silico data. I've actually learned quite a bit about the use of in silico data over the past week, so we're going to continue to evaluate this, but based on our initial work, we wanted to look at this criteria specifically. The brain malformations VSEP decided to exclude these criteria due to SIFT and polyphen-2 being more applicable to genes associated with a loss of function mechanism. And all of the genes that the brain malformations VSEP focused on were due to a gain of function mechanism. However, there are newer in silico programs that have been designed that are more broadly applicable regardless of the disease mechanism. And there's been some publications that indicate that some of these newer in silico programs may be more broadly applicable to any disease mechanism. So we wanted to start with REVEL, and so we wanted to look at REVEL and see how it would work for diseases associated with somatic mosaicism. So to do this, we analyzed the REVEL scores for 130 variants put into ClinVar as somatic for 8 genes known to be associated with a gain of function mechanism. And you can see that, as expected, the REVEL scores for the pathogenic or likely pathogenic variants were much higher than the variants submitted to ClinVar as benign or likely benign. And this difference was statistically significant. And then the variants classified as VUS were somewhere in between, and this makes sense because you would expect this category to be comprised of variants that are truly pathogenic as well as variants that are truly benign. So between this work, as well as some other recently published work on REVEL, we decided to go ahead and utilize PP3 and BP4 for our criteria. Again, it's the supporting level criteria. It is the lowest level of criteria. Next, I wanted to switch gears a little bit and focus on the criterion that we added. It's a strong criterion for genes associated with a gain of function mechanism in which a truncating variant occurs in an inhibitory domain. So let's dig in a little bit further. Again, intended for genes associated with gain of function, which we define as a gene in which the increased function of the encoded protein is associated with the disease. The truncating variant must occur in an inhibitory domain, so loss of that inhibition then leads to the gain of function of protein. In order to apply this criterion, there must be several other pathogenic or likely pathogenic variants in this region that reach that classification without the application of this criterion to avoid circularity. So let's see how this functions in practice. So this is the variant that we started with, the tech nonsense variant. It is in the C terminus. It does occur in the kinase domain, which loss of that domain then leads to the increased protein function. There are several other pathogenic or likely pathogenic variants in this region. So in this case, this PS-reg new criterion is applicable. As I mentioned earlier, we have detected this variant in an individual with a vascular malformation, and we detected this variant at a reduced variant allele fraction in their vascular malformation lesional tissue. So we've adapted the PS2 criterion to be applicable in cases like this. Additionally, as I mentioned, it's been reported in the literature in an individual with a vascular malformation, so we've adapted the case counting criteria, or the PS4 criteria, to be applicable. And as I mentioned earlier, there is the functional study, which we counted, as well as the absence from the population database. So according to the brain malformations VSEP guidelines, this variant would be classified as a variant of uncertain significance, but based on the modified framework that we've created, this variant would be classified as pathogenic, which would probably be more appropriate for this particular variant. But again, this is just one variant. How does this work more generally? So far, we've classified 64 unique variants. 69% of these are classified as pathogenic, and only 8%, or just 5 variants, are classified as variants of uncertain significance. Just of note, VUSs are underrepresented here because the vast majority of VUSs we detect via our assay are germline in nature, and so thus not classified by this particular framework. But how well do these classifications then match with what we would expect in terms of classification? So of the 64 variants, 52 were in ClinVar, and of those that our framework determined were pathogenic, 6 were in ClinVar as conflicting and 34 as pathogenic or likely pathogenic. A similar trend was seen with the variants our framework classified as likely pathogenic. And of the 2 variants that we classified as VUS that were in ClinVar, 1 was in ClinVar as a VUS and 1 was in ClinVar as pathogenic or likely pathogenic. So overall, the concordance is very high. The rules are performing as intended. So with that, I'd like to end by saying this framework for classification of variants associated with disorders of somatic mosaicism can be adapted for any disorder of somatic mosaicism, but we are going to continue to evaluate this framework. As I mentioned, we're going to continue to evaluate the in silico programs, and I've gotten Rachel's contact information so that we can work together on this. And finally, we have a ClinGen approved gene curation expert panel that was just approved, so we plan to continue to work to get this into the ClinGen framework. And with that, I'd like to thank the Clinical Genomics Laboratory and thank you all for your attention and for staying for the last session. Thank you very much.
Thank you, Alexa. We're going to take some questions now if anyone from the audience has any.
Nonsense mutations, and you may want to specify the location of that nonsense mutation, whether that's going to lead to nonsense-mediated decay, and otherwise use that specific code might be misleading.
100%. Yeah, I didn't have time to get into all the nitty-gritty details, but that's definitely something to keep in mind, whether the gene is due to loss of function or a gain of function, where the variant occurs within the protein, if it would lead to truncation or not, if it's in an appropriate functional domain. Lots of things to keep in mind, for sure. Thank you. Yeah.
And that's a great talk. I'm also working in some of this malformation, but germline is a Lois's disease. I figure out these kind of tyrosine kinase changes are very gentle. It's not like cancer. We see that keep doing things all the time. That you need to think about these people need to be survivable to be born, have some kind of malformation. It's just like the signaling happens in the wrong place, wrong time. So it's very subtle changes, feels like.
Agreed, yeah. So yeah, that's exactly why the oncogenicity guidelines and some other guidelines that have been more applicable to cancer really aren't applicable in these situations for lots of reasons, yeah. All right, thank you.